Today we'll be discussing how royal jelly's inhibition of DNMT3 is an essential component for queen development in female Apis mellifera. Apis mellifera, or the European honeybee, is a eusocial species that lives in large, complex colonies with a highly organized division of labor and caste differentiation. Colonies consist of a single queen, male drones, and female workers. These bees are essential pollinators for many crops and are often domesticated for pollination services, honey and wax production, and scientific study. Understanding Apis mellifera is important for conservation strategies that support ecosystem health and food security. Not only that, but honeybees are ideal for studying social behavior, and their advanced navigation abilities make them valuable for research in neurobiology, learning, and communication. Today's focus will be on studying factors that influence gene expression and development. More specifically, the epigenetic mechanisms driving caste differentiation between female workers and the queen. Epigenetics can be defined as the study of how cells modulate gene activity without actually changing anything within the DNA sequence. It explores the ways in which environmental and genetic factors essentially turn on or turn off a genome. Female workers and queen honeybees develop from larvae that are genetically identical. However, these genotypes can be expressed as either queen or worker characteristics. This is called phenotypic dimorphism. As you can see, workers and queens show differences in their morphology and their behavior. These differences demonstrate genomes either being turned on or turned off to create these two wildly different castes. One environmental factor thought to influence caste differences is the larval diet. During the first 3 days, all larvae are fed royal jelly, a protein-rich, nutrient-dense secretion from nurse bees. After these 3 days, worker-destined larvae are switched to a less nutrient-dense diet, while queen-destined larvae continue receiving royal jelly. This shift in their diet is where the worker and queens begin to diverge from one another, prompting research into its epigenetic role in caste determination. 3 key epigenetic mechanisms are suggested to play a role in female honeybee caste differentiation, but we'll focus on DNA methylation. DNA is a set of instructions telling cells how to make different types of proteins, and it's stored within the nucleus in the form of chromosomes. Every cell in the body contains all of the DNA instructions, but since not every cell needs every protein, the sections of DNA that aren't needed can be turned on or off. This section of DNA is called a gene, and it's specialized to make specific types of proteins. DNA is organized in chromosomes by wrapping around histone osomers, which forms units called nucleosomes. And to express a gene, the DNA must be unwound from the histone for transcription. However, DNA methylation can block this process, resulting in a silenced gene. This happens when a DNA methyltransferase enzyme called DMNT3 adds a methyl group to the cytosine of a CpG dinucleotide. When this happens, transcription factor proteins aren't able to bind to DNA, which prevents RNA polymerase from copying the gene during transcription, resulting in the gene being silenced. So, given that DNMT3 plays a major role in DNA methylation, and that there seems to be a unique component of the diet involved in these developmental changes, my question is, how does royal jelly influence the activity of the DNMT3 enzyme in the development of queen Apis mellifera? In 2008, Kucharski and colleagues had a similar question. They utilized a mechanism where short segments of RNA called small interfering RNA bind to mRNA to stop a protein from being made. Newly emerged larvae were either injected with DMNT3 siRNA to specifically target and silence the expression of DMNT3, or they were injected with a control gene. Among the larvae that received siRNA, DNMT3 levels dropped substantially after 48 to 50 hours. This time frame coincides with the 3rd day of larval development, which is considered the critical period for whether a larva develops into a queen or a worker. So, since silencing DNMT3 seems to mimic the effects of royal jelly, we can infer that royal jelly might inhibit DNMT3. The larvae were left to develop into adults and assessed for queen and worker traits. Figure A shows the number of adults in each phenotypic category. Of the larvae treated with DNMT3 siRNA, 72% developed into queens and 28% workers. The control group had 77% of the larvae develop into workers, and 23% have queen-like characteristics, but were not necessarily full queens. This difference is highlighted in Figure B, which illustrates that queen-like larvae from the control group had greatly underdeveloped ovaries compared to the DNMT3 inhibited queens, who had a far greater amount of ovarioles per ovary. In Figure C, the ovaries in the siRNA-induced queens are almost identical to those of natural queens reared on royal jelly. From this, Kucharski and colleagues' work supports that DNMT3 silencing in larvae mimics the effects of royal jelly during larval development and as an adult through the high expression of queen morphology. She and colleagues conducted a similar study where they fed larvae royal jelly for the span of either 3, 4, or 5 days, and then measured DNMT3 levels within the specific gene dynactin P62, a site that is suggested to be a specific target for methylation to occur. They found that the larvae who were fed for the longest of 5 days on royal jelly had significantly lower rates of both DNMT3 activity and expression, as well as less methylation within the dynactin P62 gene. This group produced a far greater proportion of queens and a significantly lower proportion of intercasts or workers. This work supports the notion that royal jelly's inhibition of DNMT3 is a key driver for queen development in Apis mellifera. By inhibiting DNMT3, Kucharski and colleagues demonstrated that silencing this enzyme can sufficiently induce queen-like development even without a royal jelly diet. Further evidence by Shi and colleagues, larvae fed royal jelly for 5 days showed significantly lower DNMT3 activity and gene expression, along with reduced methylation at the dynactin P62 site. These larvae also produced the highest proportion of queens and the fewest proportion of workers or intercasts. Overall, these results suggest that royal jelly likely suppresses DNMT3 activity, leading to reduced DNA methylation and the expression of queen-specific genes. These findings are significant on many different levels. On face value, Kucharski and Shi's research provide a deeper understanding of the molecular basis by which caste differentiation occurs, opening the door for further advancements in honeybee biology. In terms of honeybee management, this finding could inform new queen rearing techniques in order to control the queen population, which is essential for the colony's health and ensuring that our crops have enough pollinators. On a broader scale, these findings confirm the use of Apis mellifera as a model organism for studying epigenetic mechanisms, even in humans. Overall, this evidence strongly supports that royal jelly drives queen development in Apis mellifera by inhibiting DNMT3 activity, and these findings have implications across a spectrum of fields, from beekeeping strategies to cancer research.